Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to Day 3 of the MDDI QMed webinar series, Urgent Update on ISO Standards, Changing Safety and Toxicity Compliance, sponsored by Nelson Labs and broadcast by Informa. I'm Daphne Allen, editor of MDDI, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media outlets, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. You may also download a copy of the slides via the resources widget. Toward the end of our webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a minute to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. Lastly, if you're experiencing any technical problems, please click on the Help widget found at the bottom of your screen, or type your issue into the Q&A area, and we'll be glad to offer one-on-one assistance. Now, onto the presentation. Cleaning, disinfection, and sterilization of medical devices. Updates to reprocessing guidance documents AAMI TIR12 and TIR30 ST98. Discussing today's topic are Alpa Patel, Principal Scientist for Nelson Labs, and Geraldine Franson, Associate Technical Consultant for Nelson Labs. To learn more about our speakers, visit the bio widget. Alpa, I now hand it over to you.
Hello and welcome to the final day of the webinar. Today we are shifting the focus from toxicology and biocompatibility to hospital processing. My name is Geraldine Franson and I will be discussing the updates to TIR12. My associate Alpa Patel will be sharing the new ST98 and updates to other processing standards. The guidance document TIR12 was published in December of 2020, so it is a fairly new document. The organization and information in TIR12 was updated and expanded with new sections, which include Section 8, Risk Analysis and Management, and Section 6, Risk Analysis and Management, and Section 8, Presentation of Information. A lot of the language and information is the same between the two documents, but the order it was presented in has been shifted around and updated to better tell the story of hospital processing. And as a writer, I'm a firm believer in telling the story as well as you possibly can. We are going to start with Section 4, Device Design Considerations, which was previously Section 3. This section was expanded to include A, risk of patient infection based on clinical use, G, packaging design applications, considerations, E, cleaning, H, disinfection, and I, sterilization. Remember, as a device manufacturer and designer, it is key and critical to consider these aspects during device design rather than discover the hard way that the material you've selected is not compatible with the sterilization method you want to use. Please learn from, the experience of the writers of this document. All right, we are going to begin with Device Design Consideration A, risk of infection based off of clinical use. And before we discuss the risk of infection, we're going to skip ahead in the document to Annex F. Table F1 is a new addition to TIR12 and shows really resistance to sterilization and disinfection and microorganisms. At the bottom of the table, you can see our microorganisms such as lipid envelope viruses that pose the least challenge to inactivation by sterilization and disinfection. And I use inactivation because viruses are not alive and can't be killed. At the top of the table, we can see bacterial spores, which are going to pose the greatest resistance to inactivation. Technically, all of the microorganisms on that table could pose a risk to the patient if introduced into normally sterile areas of the body, but we use the most resistant to sterilization bacterial spores in our validations to demonstrate that if we can inactivate bacterial spores, we can also inactivate all the other microorganisms on the table. On the other end, at the bottom, the lipid envelope for the microorganisms that would pose the greatest risk to infection for intact skin would be your normal, everyday vegetative bacteria and lipid envelope viruses. So we use 4 species of vegetative bacteria to demonstrate a low-level disinfection. If blood-borne pathogens are a concern for infection with devices that contact intact skin, an intermediate-level disinfection using Mycobacterium should demonstrate that you can also inactivate any pathogens that could cause an infection. The previous version of TIR12 focused on patient contact and the Spalding classification to determine what level of disinfection or sterilization was needed. So if a device contacts sterile areas of the body, that is a critical device, and sterilization is required. Likewise, devices that contact mucous membranes or non-intact skin are designated as semi-critical and ideally would be sterilized, but if sterilization is not compatible with the device, either due to materials or electrical components, a high-level disinfection can be performed. And then finally, devices that contact intact skin or have no contact with the patient are designated as non-critical and need either a low-level disinfection or an intermediate-level disinfection. However, there is a large hazy gray area in determining which non-critical devices needed intermediate-level disinfection or low-level disinfection. The new update to TIR12 changed the focus from contact to the patient to the risk of infection to the patient. If a device can be soiled with blood or other potentially infectious materials, then at minimum the device should undergo an intermediate level disinfection. And from an infection perspective, this makes a lot of sense. It doesn't matter who's touching the device. The key factor is what it could be contaminated with, because if a device contaminated with the blood is directly contacted by the patient, the infection risk is the same as if the device could be contaminated with blood and is touched by the caregiver or provider who then touches the patient. Annex F in TIR12 also offers further guidance on when an intermediate level disinfection or a low level disinfection should be performed, with suggestions based off of parts of devices and medical equipment that receive high levels of touch and interaction during use. But there's still some gray area as to when intermediate or low-level disinfection should be performed, but now we can see that there are some visible edges between the shades of gray as to what should be performed on which devices. Moving down the list of device design considerations is D, human factors, which was previously titled Misuse-Related Design Considerations. It is important to remember that the processing steps listed in the instructions for use are going to be performed by a human being, and human beings are very imperfect creatures, including myself. We are all prone to mistakes. In addition to a detailed instruction for use, manufacturers are encouraged to provide on-site training and education for the sterile processing personnel. This opens communication and collaboration on the device designs to help improve the device designs that meet the functional requirements in the risk analysis, and also addresses human factor considerations. Related to human factors is the new Section 8, Presentation of Information. IFU developers need to design the instructions to clean and process devices with the end users in mind. The FDA guidance document also contains a section specific to the contents of IFUs and outlines what the reprocessing instructions should encompass and how they should be presented. Additional examples of how processing instructions could be presented can be found in Annex D and Annex E of the updated TIR12. This section suggests that all instructions should be grammatically correct, legible, and presented in a logical order, from the initial processing step at point of use through the last terminal reprocessing step. And each step should be well written and simple in language, with as simple language as possible. Last on our list of design considerations is thermal disinfection and steam sterilization. Thermal disinfection is outlined in Annex F, and it's important to remember that there is a distinction between thermal disinfection performed as an interim step, which is required in the European Union, and thermal disinfection done as a final step prior to using the device on the patient. Notable updates to the steam sterilization section were the removal of the dry times acceptance criteria. For additional information on dry times, the readers are referenced to the new ST98 and the ISO document 17665-2. And then finally, a full cycle approach was added to give sponsors additional flexibility in how steam sterilization validations are performed. And for my fellow math and data nerds out there, we've included some beautiful math showing that the sterility assurance level achieved using both approaches is the same. Thank you for your attention. I'm now going to pass this over to Alpa Patel to discuss the updates to the new SC98.
Good afternoon, everybody. This is Alpa. I am going to be speaking on the second half of the presentation and go over the mammoth project that we have been working as a committee in the AAMI regarding TIR30 turning into standard. This has been going on for a couple of years, and I think we're in the final stages. We're going to have a meeting next this month, actually, to go over the final comments that we received on this document. Now, this document is for cleaning validation of healthcare products, and that includes all reusable medical devices in the market, and goes over all the test method validations and requirements that are needed for submission to the FDA. Now, what is updated or new in ST98? There are certain things that you are already used to seeing when it comes to reusable medical devices. However, there are additional things that the committee has decided to add in or elaborate on or clarify more further. And some of these things I'm going to go over in detail as we go through the slides. However, these are some of the bullet points that we will touch on, and I will encourage any questions or follow up on any of these things if I haven't gone through it in detail. Addition of controls with detailed explanation. The standard has adopted some controls in this final draft. Addition of simulated use cycles, which was in the FDA guidance document for validation and labeling for reusable medical devices. However, nothing out there had any information on it, but this standard that will come out will have it. Addition of 2 analytes for testing, also increasing the controls. And acceptance criteria regarding results. How are the results going to be presented now as they have been historically? What does non-critical devices look like in the world of reprocessing? This has been a topic that has been a burden on my side for a very, very long time, and I'm glad to hear that finally we have come to a resolution that makes much more sense in the practical life of clinical setting than something that was just done because it was in a standard. And applying test results, how are they going to be identified and looked at, and how are we going to evaluate those? And there are some criteria that have been applied to these that will be new to this industry, especially for reusable. They are applied in different industries, but not for reusable devices. But now we're going to apply standard deviations to it and see how the test results are similar to each other. So they are quite big changes, but changes that we did foresee that will happen in a few years, that things will be addressed, but nothing was written down but thought of. But finally, TIR30, which is a technical information report, so not a standard, but is going to be converted into a standard. It's still in the draft forms. It's been for, I think, 3 years now, but we're right there at the end of the process. Now, for controls and validation, I'm sure if you have been working with reusable devices, you are aware of these controls and have been testing them since 2015 when the FDA guidance document came out. They did label all these controls in there, but it was only in the FDA guidance document. And when it's a guidance document, it's not something that you have to do, but it is a recommended practice that you should consider. And because it was considered in that manner, it made sense to add it to a standard that talks about cleaning validations for these devices. So they were actually taken out of the FDA guidance document and imported into this in this standard. There was a lot of discussion regarding what each control means, but we kept it the same as what the FDA thought process was, because it did make sense. I'm going to go through the controls, not in detail, but just give you an outline of what these controls will look like. There is a negative device control. Now, this is just a control that shows you that once your device has been processed through your chemicals for cleaning, there is nothing left on there that can introduce contaminants during the assay. So it is a control that shows that the detergents were removed through a rinsing process properly and did not interfere with any of the test results. The positive device control, of course, is a device that is soiled and tested side by side with the test samples, just to it gives you a starting assurance of what the soil components are on this product after soiling, and after reprocessing it, what is left over. So this is a control. It's a test method control, and it's a required control for sure. The negative sample control is just blank, basically. It's just a blank to show that the equipment and supplies that were used during the processing of these devices were free of any contaminants that would interfere with any of the analyte assays that we're going to use. So this is a good control. We already have this control in place. So it's not something new. If you are in the validation industry, it's used everywhere. Even if you're doing chemistry, or if you're doing reprocessing, or you're doing biocompatibility, this control has always been there. Now, positive sample control is a new control that's been there, but not relatively discussed openly. And this control is one that is assessing the test soil that you're using. As you know, for if you're not aware, for cleaning validations, we're using simulated use test soils. Now, these test soils can be purchased or can be prepared in-house. And when you prepare in-house, you have to do some qualifications, and to make sure that if you're testing your end product with protein or hemoglobin or TOC, whatever you are assessing can be picked up by the assay that you're performing for your end result. So this control is in place to show that the test soil that you use can be picked up by any of your assay, and the amount of test soil is close to the LOQ of the assay. So you know that it was there, because what we're looking at is if after you've cleaned the devices, you will see your extract fluid. Can the assay pick up low levels or trace amounts of soil that's there? And that's what this control is actually establishing for you. So very important controls to have in a validation process, and historically they have been used, but not to the point where it was something that all test labs did. But it was something that was in the FDA guidance for a while. This next slide goes over simulated use methods, also coming from FDA guidance document in 2015. There was a little information on it, however, nothing was elaborated. It wasn't expanded. It was left to interpretation of the industry to figure out how to perform these validations. And this new standard has helped us define those guidelines and pushed the line to what is simulated use methods. And what do these cycles look like? What is the need for these cycles? And what you want to understand with these cycles is that you are processing them through how it would be processed in a clinical setting. So you're just putting it through patient use, which is soiling, and all the cleaning processing that will happen on that product. So if it's cleaning and disinfection, then that's what you're assessing. If it's cleaning and sterilization, that's what you're putting through the simulated use cycles. Now, this is a must for critical and semi-critical devices. So anything that will be punctured through the body into the bloodstream, or anything that is going into mucous membranes, simulated use cycles is a must. The only point where you can play around with this situation is when you are in a non-critical device zone. That's your safe zone, where these are devices that are only in contact with intact skin. Those may not require simulated use. However, like Geraldine said, there is a gray zone between the non-critical devices. You have to assess for risk of healthcare acquired infection, and if it's low, then you can say that, well, we don't need it. But if it's higher, and you say that there's a lot of back and forth with the user when they're using this, so maybe we need to do the simulated use. So it is still it is still left on the table. However, the standard does say for non-critical devices you don't have to perform this. Now you might ask, well, how many cycles do I need to perform of this? What is a good cycle number? So in the previous versions of SC98, we did put a cycle number. However, the committee during the final drafting decided that no, we're going to leave that. We're going to leave it to interpretation. So number of cycles is not written down or given a number. However, 6 simulated use cycles has been accepted by regulatory and has been used historically for all reusable medical devices since the 2015 guidance document came out. So we would recommend at least do 6 cycles of simulated use cycles for critical, semi-critical before moving on to the cleaning validation. Now for positive controls, that is another update, is there was nothing specified in documents that you needed 70% extraction efficiency for your positive controls. Now we do have that. It is in the guidance, it is also in the ISO, and they are moving towards that. You can't have a low extraction efficiency. And 70 has been historically thrown out in USP, in ASTM, so you will see, when you see this number, you will resign with the number like, yeah, this is common that we see. But it wasn't established for reusable medical devices. They didn't extrapolate to that. But now we have that, and you will have to meet greater than 70% extraction efficiency, where you would apply this to your test samples as well. Another thing for the positive control is the reusable industry has always been okay with doing just one positive control with the test sample. It is required that you need a minimum of 3 positive controls now. And like I say, the language says shall be used, so that means you will have to use at least 3 positive controls. However, in some instances, a smaller size can be used, however, will have to be scientifically justified. I'm sure you've seen this language in many, many documents. It's a common language that you see. And when would be a case where you would justify this? And from my perspective, when I read this, I would think that it would be for non-critical devices, devices that require low-level disinfection that are non-patient contacting devices, keyboards, iPads, things that are in the OR room or in a clinical setting where they are not touching the patient, and so, but we are still doing reprocessing instructions for them. In that case, it could be justified that you can do 1 positive control because it is non-patient contacting. The risk of infection is very low, and also we don't need to worry about doing so many controls because the test soil accumulation over time is not there. Now this is a big one for all of us. This is where it was established where the test replicates that are being performed, and you will see there's 6 test replicates here as sample size for test articles is also going to increase. This is just an example though from the standard, and this example is in the final draft as well. What this example is stating is if you look at the example 1 right on the top, you will see that there are all these different values that you receive. For current state, these, for analytical people like us that are in the lab, from this we can see, well, there's something off about this, but we just need to show a 6.4 micrograms per centimeter squared for protein. So does it fit the acceptance criteria? Did it make the acceptance criteria? Yes, it did. But the differences between the replicates are quite significant. They're different. So that is something that would be investigated and looked at. However, it wouldn't fail the test, right? So now it is requiring that you would have to perform a standard deviation calculation between these test samples, and you would have to apply the standard deviation value to the highest test result you received. And if that is higher than the acceptance criteria, then you fail the test, and you would have to repeat the test again with a higher sample size. So now that you have 6 here, you would have to do it with an increased sample size. And when we think about increased sample size, we never think 1, 2. It's always in triplicate. So we would have to do 3 extra. It is my interpretation, not everybody's interpretation, but this is how I would interpret it. But this is a big change for us in the industry because we've always just relied on the acceptance criteria that is out there and said as long as it meets that acceptance criteria, we're good to go. Now, cleaning validation acceptance criteria, I just wanted to go over some things that have been added to the final draft for ST98 and ISO 15883-5 if you're not aware of it. We did add TOC and ATP to the SC98. The current draft of TIR30 doesn't have TOC or ATP in it, so this standard will have that. They are all the same as they were before, and for TOC they added the little less than or greater than 12 micrograms per centimeter squared, less than or equal to 12 micrograms per centimeter squared for TOC. And this value was researched a long time ago by FDA. They presented the data in a webinar in 2005, believe it or not. And that value has stuck in the industry for TOC. The ATP usually are not used for cleaning validations. They're mostly used for R&D or something that you would. This is what healthcare facilities would use to assess their processing steps. It's a quick study where they give you a value in RLU, which is relative light units, and per test kit of that, they will tell you what to multiply with to get the femtomoles per centimeter squared. But that is a value that you should achieve. But this I would see more for R&D work or healthcare or just to see where you're at. This is the benchmark you should be at. Now, the ISO 15883-5 has significantly impacted the cleaning validation by adding the action and alert levels. We know that the FDA does not care for these alert levels. However, we don't know how other regulatory efficient agencies in EU or other countries would, but these are some of the alert levels that have been added, and where they came from, it was mathematically just calculated and put it in. I don't think there is substantial data gathered, but for protein there was, but not for the rest of them. So this is something that you may want to be aware of. Now, if you might ask me, what does alert mean? Is that going to be an action item? I think the action item is only if you're greater than 6.4. Like you have to be greater than all of these benchmarks where action item will be addressed. Alert levels is something that when you are drafting your IFU and you are doing these validations as an R&D and you're getting these, this will give you an alert like we are halfway to what the benchmark criteria is. So maybe we want to look into adding more processing parameters or figuring out another test method to establish these. But just so you know, this is something that you want to be familiar with for sure. Now, on the bottom of the slide, you can see that I've written for critical and semi-critical devices, you want to have a visual inspection. That is a must with 2 quantitative analysis. So you have to test at least 2 markers out of this panel that I presented here on the slide. The most common is protein. You have to do protein. It's a must. I don't think that is negotiable. And then you would go down the slide where it says hemoglobin, TOC, carbohydrates. So I have ranked it between how it should be considered for testing. Now, for non-critical devices, this is a new for everybody. This is brand new for the industry. It says visual inspection only is needed for non-critical devices. Now, baseline acceptance criteria. So, like we looked at the one before here, you see that there is a 3 micrograms per centimeter squared. That is where a lot of this is coming from. This standard in Germany has the sorry, I just spaced for a minute. Germany has 3 micrograms per centimeter squared. The RKI also has, that is another one in Germany that has 10 micrograms per instrument. So for protein, they are different numbers. So I just put the slide in as an awareness slide that there is just not one value for protein. You have to go and figure out for countries, they might be different. We know for Germany, they do look for 100 micrograms per instrument. At certain notified bodies will ask for that, but certain will be fine with just the 6.4 micrograms per centimeter squared. Like I talked about for non-critical devices, visual inspection alone is sufficient for cleaning validations, so that when the standard is finalized and published, it will be in Section 8 in the endpoint section. And it is something new. It is something we have been waiting as an industry for a very, very long time to happen. So I'm very excited that finally it's got the attention that it needed. Along that lines, non-critical devices, a new segment of ISO 17664-2 was published this year, and this document only focuses on non-critical devices. And in this document, it does say in 6.2.1, you'll see at the last sentence, disinfection may be carried out concurrently with cleaning of the medical device. And so it does allow for certain products to be as a combined process. That's what we would call it, a combined validation where cleaning and disinfection can be performed in one step. Now you may say, what does that look like? Like Geraldine said, there is your gray zone, but we're finally identifying these zones and trying to give it some light. And that is something that you would have to talk amongst your colleagues or even a third party to see where you fit in that category and how we can help distinguish if they can be performed together or they have to be performed separately. Of course, there are some guidelines that you have to follow for that, so you would have to evaluate those as well. Now, ISO 15883-5 is still in draft phases, but the 2020 version of the draft phase also says right here on the bottom that non-invasive medical device shall require visual examination only. So you're seeing that the industry is moving in the 3 documents that I just presented, moving towards that non-critical devices can be fine with just visual examination only. There's no need to do protein, POC, or protein hemoglobin on these type of devices, as it's not relevant in the healthcare-acquired infections coming from these devices are not high at all. And you can also see that it does say for invasive medical devices, at least one other validated method shall be measured in addition to protein. So you can see here where protein is definitely the marker that the analyte that you want to test, but you can test something else with it. So this is an ISO document which is used worldwide, and the AAMI document has the AAMI ST98 document also has that in there. So we're now finally aligning all the documents together. Now, a big change that's going to happen is regarding the ASTM2314. Historically, a lot of international, not US, US doesn't cite this often, but we do see Europe, Japan, Australia. They cite ASTM2314 as it's been in the industry for a very, very long time, but it's very, very outdated. So we will be retiring that in the ASTM and adding it to another document that we have published in the committee, and that is going to be a standard guide for detection of quantification of cleaning markers. And I was assigned to do that, put that transition, so we will be doing that this November. End of life use testing. This is one that's quite in discussions and everybody is talking about, is do I have to do it or do I not have to do it? What is the question? So here you will see conflicting text and comments on it. However, what I have seen in the industry is if you write in your IFU where the users can identify that this device can no longer be used and how they can identify, you can justify outdoing the end-of-life use testing. Now, ISO 10993, this slide right on the top, section 4.8, does specify for reusable medical devices, biological safety shall, as you all know, that means you have to be evaluated for maximum number of validated processing cycles by the manufacturer. So because there's a shall there, that mandates that you should have that information. But if you look at the MDRs, I have it here, it does say that if the device is reusable, information on appropriate process for allowing reuse, including cleaning, disinfection, all of those things, information shall be provided to identify when the device should no longer be reused, signs of material degradation, or maximum number of allowable uses. There's an or there. So if you read between the lines, there's an or there that the MDR does allow that as long as you have or have written how you can identify this degradation or how it cannot be, how it is not usable anymore to the users or the processing person that is actually processing these devices in your IFU, you may not have to perform these cycles. ISO 17664 also says that, and the FDA guidance document also says that. MDA must determine if reprocessing leads to a degree of degradation that will limit the useful life. MDM must determine the number of reprocessing cycles or on the indication of the end of its life. So you have to have some sort of language in the IFU. Either you write the number of cycles, which you will have to validate and show if you write in the IFU, or you have to determine at what point does my device degrade and at what point will the user know that this device is no longer usable. Those are the questions that you have to answer. So there is an or there. But we have been seeing a rise in these life cycle testing, and because there is more and more inquiry on it, and regulatory is asking more and more information on it. If you say, well, it degrades after so many times, or it discolors, they may challenge you and say, how do you know that? What test did you perform to establish that user will notify? Can you please go over that test? So there are leeways that you can fall, but there are some things that you can also take into consideration there. Now, these are some of the standards for reprocessing that you may want to be aware of that are up in Horizon. A lot of people know about the AAMI, they know about the ISO, but they fall short in the ASTM side of things. And ASTM has been working alongside with AAMI and ISO to come up with two new standards to explain more detail to what AAMI and ISO specify. AAMI and ISO usually give you guidelines. ASTM gives you test methods. So if you're not involved in any of these, you may want to be involved in these committees, and that gives you more information on how to perform these validations. That takes me to the end of my presentation, and our contact information is on the slide. So if you have any additional questions or you want to talk further about what we have presented, we are happy to do so with our contact information. And for more educational events and additional information regarding our training, webinars, seminars, and other related information, you can get information from these websites that are on the slide, and can get more insight on what we do as Nelson Laboratories and Sterigenics. And this takes us to any questions. Please submit your questions via the text box below, and we're happy to answer any questions you have. Thank you for listening to both Geraldine and I regarding the updates in reprocessing. It is the time right now for reusable medical devices, and things are changing dramatically in the platform for these devices, and a lot of changes are up ahead coming as well.
Great. Thank you, Alpa, so much. And thank you, Geraldine, earlier for your presentation as well. So as Alpa mentioned, it's time for our Q&A session. And so as a reminder to our audience, to participate, just type your question in the text box located to the right of the presentation window, or click on the Q&A icon at the bottom of your screen. If we're not able to answer all submitted questions during today's webinar, we'll be sure to share your questions with our speakers who can reply offline. So let me jump in right now and see. I think our first question actually is for Geraldine regarding, I believe, AAMI TIR12. And the question is, are implantables lumped in with critical, or is there a higher standard for implantable devices?
For reprocessing implantable devices, so these would be implants that would either be processed before use on a patient, or sometimes implants will be, like for tooth implants, there might be a range of sizes, and then the dentist will pick which one is appropriate and then clean the other ones. They're critical devices. They're contacting those sterile areas of the body and need to be processed accordingly, although most implants are only processed once, but sometimes they can go through multiple cycles with a good justification.
Okay, excellent. Thank you. Alpa, when is ST98 going to be published by AAMI?
Very good question. So that is something that is going to be discussed in the upcoming meetings in August. We have 2 meetings scheduled in August to discuss the final comments that were received for the draft. We received 260 comments, so we're going to go through those. And then once the comments are resolved, we can see what will happen in the next process. We're thinking it will probably be end of this year, or hopefully Q1 of next year is our hypothesis. That's where we're going to be published.
Okay, excellent. Thank you. Here's another question. Are there standards or guidance documents that specify the types of water to be used in a cleaning process, i.e. deionized, critical, tap, etc.?
I can take that one as well. Yes, TIR34 currently is quality of water processing for reusable medical devices. So a lot of standards do reference to AAMI TIR34 is water for processing. But if you look at FDA guidance document, or if you even look at the ISO documents, 17664, ISO 15883, and also the current TIR30, which is not converted into standard yet, that does go over what each phases should be used. Commonly, for reprocessing, when it comes to cleaning validations, the first few phases are done through tap water. That's what the hospitals use anyway. So that is what's used for the validation. But the final rinse before it goes to disinfection or sterilization is done with treated water, some sort of treated water, and that's usually how it's processed.
Okay, great. Thank you. With the upcoming changes in reporting acceptance criteria for cleaning validations, how will this affect our legacy products or products that were validated before the standard will be published?
So that's a very good question. We have a lot of legacy products out there. We have products that were validated a few years ago. With this new requirement where you have to apply the standard deviation to the test replicas and apply to the highest test result, this is something that that you would have to go back and make sure that the test replicates that you received for your product were within the specification. Yes, it will take more work, but this is something that the quality assurance departments or even our consulting team here can help with. We are aware of this process. We are aware that this is going to come down, and we are aware that these questions will arise at some point to assure that 5 years ago you did a validation on this product. Did you assess the test results? And we can definitely help with all of that as well. So you have a couple of options there, but it is something that you would have to go back and look at.
Thank you. How does the life cycle testing play a role in reprocessing, and is this test required?
So with life cycle testing, like I said at the end of my presentation, ISO 10993, the language used shows it's required. However, there are other documents that do specify that as long as you inform the user in your IFU how to identify where they can figure out that this device is no longer usable, then you are fine with it and you don't have to perform the cycles. Now, do I feel like this will be sufficient enough? I think for now, because they have that language in the documents, we can always reference that, hey, this says that as long as I show how the users can identify it's no longer usable, that's good enough. But with the ISO 10993, we are seeing more and more reuse cycling to assess for biocompatibility for patient care, so and patient safety. So it might be something different on that end, but it's still an open-end question at currently.
Is FDA going to ask for alert and action levels for cleaning validations once ISO 15883-5 is finalized?
So I went through that as well on my slides, and FDA does not recognize the action and alert levels, so I don't think we would have to worry about that for FDA submissions in US. However, in Europe, it is something that we are not, it's still in the draft phases, it's not been published, so, but it is going to be published this year. So how the Europe will react to that, the notified bodies, that is still up for us to see if they are going to be holding you to the alert values or the action values, or how you would have to address if you had either one.
Okay, great. Well, thank you, Alpa and Geraldine, for answering questions for our audience. Really appreciate that. And especially thank you for your presentations today. So that's all the time we have for our questions. Definitely want to thank our sponsor, Nelson Labs, as well as to everyone in our audience. We appreciate your attention and participation. Within the next 24 hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues or peers who may not have been available to listen to our event today. This webinar is copyright 2021 by Informa. The presentation materials are owned or copyrighted by Nelson Labs and MDDI QMed. The individual speakers are solely responsible for their content and opinions. Thank you, Alpa and Geraldine, very much.